everyone, my name is Dewi Syahidah and I am a research staff at IMRAFE Gondol, Indonesia. Today I would like to share with you about PCR column contamination gave false positive results for Cherax quadricarinatus ambidensovirus in Spodoptera frugiperda cell culture. This is the outline of what I'm going to talk today. First, I would like to give you a brief introduction and then some objectives of the study. Following that, I will give you the materials and methods that we use. And then finally, I will give the result and discussion. There has been very broad interest in red claw crayfish aquaculture since the last 3 decades. Industry developers were rapidly learning the species' benefits. However, crayfish diseases have become important with several infections being described from farms in Australia since the 1990s following the establishment of the industry. Research studies on virus pathogens of freshwater crayfish have attracted much interest for decades with a growing number of published reports. A parvo-like virus was initially reported infecting red claw crayfish in Queensland, and it was described as Cherax quadricarinatus parvo-like virus or CqADV. The virus was responsible for causing mass mortalities within a farm. 13 years later, Bocho et al. 2015 examined CqADV and proposed a new species name of decapod ambidensovirus. Bocho et al. Then named the virus as Cherax quadricarinatus ambidensovirus or CqADV. A major obstacle to control the virus in crustacean farm is the lack of standards in identification, isolation, and purification of the viruses. Detection in low-level infection considered less sensitive. That was why we need PCR. However, PCR-based column is sometimes limited by contamination in the column. These 2 study groups here confirm that they experienced contamination in the columns that they use in their PCR reaction. The study of crustacean virus replication is limited by the lack of suitable cell lines. For many animals, cultured cell lines are considered indispensable tools for studying the molecular and cellular mechanism of pathogenesis. The use of cell culture models to grow viruses provides a potentially limitless source and thus facilitates its characterization. Attempts to study and the in vitro propagation of crustacean viruses in cell culture derived from insect organs have been reported. The most common insect cell cultures used are derived from mosquito or CqADV. The growth of crustacean viruses in C636 in the previous studies was still questionable and tends to be negative. Therefore, it is crucial to examine insect cell culture as alternative to mosquito cell culture. Another insect cell culture that has been widely used to study viruses derived from Lepidoptera or Spodoptera frugiperda, ovarian tissues or SF9. Therefore, in this present study, we would like to in vitro propagate CqADV in SF9 cell culture. We would like to examine the virus propagation by staining and PCR. And then we would like to examine the potential of extraction column contamination of our PCR that affected our result. Now, move on the materials and methods that we use in this study. First, we collect the CqADV inocula from frozen tissues of CqADV infected crayfish at James Cook University, Australia. And then we grow the SF9 cell culture in 75 tissue cell flasks. We use serum-free media and we add streptomycin. We divide the tissue culture flasks into two groups. The first group received CqADV inocula and we call it as the infected groups. Whereas the one group as negative control, we add only 1 time PBS. 50% of cell suspension of each flask were collected for 12 passages every 5 days and they were subjected to H&E staining and PCR. This table here illustrates the primers used in this present study based on Bocho et al. 2015. For the DNAs, we collect the CqADV inocula, the positive and the negative SF9, and also the non-template control. For the PCR amplicons, we use agarose gel electrophoresis, and we visualize the gels using a UV transilluminator. After that, we clone and sequence our samples. We send to the macrogen, and then we do NCBI blast nucleotide. Now, move on to the results that we found in this study. This figure here is the H&E staining of SF9 cell cultures at different days post-infection. The top row here are the CqADV infected SF9, whereas the bottom row here is the uninfected CqADV SF9 as the negative control. From this figure, it can tell us that we cannot see any difference between the two groups. This figure here is the PCR amplification of CqADV in SF9 cell culture using this primer set. As we can see here in this box here, can be clearly seen that all samples show positive result for CqADV, which is 280 base pair. Now, this figure here illustrates the PCR amplification of non-template control samples using this primer set. Again, can be clearly seen here that all samples show positive for CqADV, which is 280 base pair. We analyze our sequence and we blast the nucleotides against the nucleotides deposits in the NCBI website. We found that none of our sequence matched with the sequence of CqADV. So we suggest that there might be a contamination within our PCR column. These results here confirm the two previous studies by Erwin et al. 2011 and Nasasi et al. 2013. Erwin et al. Confirmed multiple PCR water controls not processed through the column were consistently negative, indicating that the PCR buffers and polymerase enzyme used in the PCR were clean. Whereas Nasasi et al. Found a potential association between a virus they test, in this case parvovirus-like hybrid virus with diatheme, that generate silica matrix used in the spin columns may have resulted in inadvertent viral contamination during manufacture. Now, We conclude that H&E stains failed to show the changes due to CqADV infection. However, the amplification of both SF9 groups were positive for CqADV. PCR amplification of all NTCs were positive for CqADV. None of the blast nucleotide results matched with the sequence of CqADV. These false positive results suggested contamination in our PCR columns. Our primer sets might not be sensitive enough to detect CqADV in SF9. A non-column PCR extraction needs to be developed to detect CqADV in cell culture. These are some references that we use in our study. Thank you very much. Any questions so far?